Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Andres Cubio Ruiz. Dr. Cubio Ruiz is a scientist at the Wyss Institute of Harvard University, an Institute of Medical Engineering and Science at Massachusetts Institute of Technology. Dr. Cubio Ruiz worked on an engineered live biotherapeutic product that safely degrades broad-spectrum antibiotics that cause major disruptions to commensal bacteria in the gut and results in dysbiosis. Today, Dr. Cubio Ruiz presents his session, Protecting the Gut Microbiota from Antibiotics with Engineered Live Biotherapeutics. Dr. Cubio Ruiz, over to you.
Hello, my name is Andres Cubillo Ruiz, and I'm a scientist at the Wyss Institute of Harvard University and at the Institute for Medical Engineering and Science at MIT. I first want to thank the organizers of this Microbiome First Summit for the invitation to present our recent work on using engineered bacteria to protect the gut microbiota from the harmful effect of antibiotics. What I want to do today is first tell you about antibiotic-induced dysbiosis and why preventing the damage to the gut microbiota is an important unmet need in healthcare. Then I will talk about how we have devised a new approach to protect the microbiome using engineered live biotherapeutics. And then I will show some proof of concept data of this technology in a mouse model. So in recent years, we have gained greater understanding of the roles gut microbes accomplish in the human body. Before, we used to think that those microbes were just there, maybe to help with food digestion. But now we know that they are an integral part of us. They are needed to exclude potentially invading pathogens. They are essential for the development and proper function of the immune system. Their metabolic activity also modulates the communication and function of the gut-brain axis. These are just few examples, and every day we see more and more discoveries about the critical roles of the gut microbes in health and disease. While it is still difficult to define what a normal or healthy composition of the microbiota is, in general, the picture that emerges from all these studies is that a diverse composition is associated with a healthy status in humans. Now, the problem comes when certain components of our lifestyle, such as medications and particular types of diets, affect the composition of our gut microbiota. Most notably, antibiotics, since they cannot discriminate between pathogenic and beneficial bacteria, they decimate the microbiota and create an altered state called dysbiosis. In here, some microbial groups disappear, some others increase their abundance, and overall, the metabolic environment of the gut dramatically changes. This imbalance can lead to serious unwanted consequences. The gut in a dysbiotic state is more susceptible to opportunistic infections, most notably C. difficile. Dysbiosis increases the risk of developing inflammatory and metabolic diseases. And it also has been shown that it can affect the efficacy of certain medical treatments, especially if they intersect with the immune system, like checkpoint inhibitor therapies for cancer. Now, the issue here is that antibiotics are life-saving medicines, and in their action, they cannot differentiate between commensal and harmful bacteria. A patient that presents with bacterial pneumonia must be promptly treated with antibiotics. If a kid has recurrent ear infections, antibiotic therapy is necessary, or you might be risking permanent hearing loss. Fortunately, most of the times antibiotics work very well and you can cure the infection, but inevitably you will end up with an underlying gut dysbiosis. In some patients, the consequences of this alteration can manifest in the short term, for example, as a secondary infection with C. difficile. In other patients, silently, in a longer term, together with other risk factors, dysbiosis can induce the development of asthma or obesity or other non-communicable diseases. But given the usual urgency with which we must treat the bacterial infection, historically, we have been accepting these downstream consequences. Now that we are realizing the importance of the gut microbiota, we need to do better to avoid this collateral damage of antibiotic therapy. And the fact is that when treating an infection, the vast majority of times, the antibiotic action is not needed in the gut. If you have a urinary tract infection, soft tissue infection, pneumonia, or even prophylactic treatments, there is no need for the action of the antibiotic in the gut. And you are all the time indiscriminately killing gut bacteria. So how this damage happens? When a patient needs antibiotics, it doesn't matter if they are administered orally or parenterally. Once they are in circulations, antibiotic will eventually reach the liver, and an active fraction of it will be carried down the gastrointestinal tract through biliary excretion. Then gut microbes are going to be exposed to the antibiotic, killing susceptible cells and inducing this state of dysbiosis. Under these conditions, the gut is more permissive to colonization and has impaired barrier function and is prone to pro-inflammatory states. And importantly, after the antibiotic treatment, the gut is enriched in resistant populations of bacteria that have survived the treatment. So for all those instances in which the antibiotic is not needed in the gut, we must spare the gut microbiota. And for that, currently, there are no effective interventions. People usually take regular probiotics during or after an antibiotic course, but the reality is that these are not truly effective, as normal probiotics cannot protect the microbiota because they are as susceptible as the antibiotics as the gut microbiota. Also, the complexity of the current probiotic formulations does not compare to the complexity of the natural bacterial populations that you build up throughout your life. And therefore, they cannot supplement the functions of the endogenous gut microbiota. In fact, it has been demonstrated that regular probiotics can delay the recovery of the native microbes. But overall, supplementation of the gut with microbes have been shown to be safe in many studies. Now, what we propose to do is to build up on this strategy, but instead of using a regular bacterium, we can use a synthetic biology approach to endow a microorganism with a specific therapeutic function, and that is protecting the gut microbes from antibiotics. And this would be what we call an engineered-like biotherapeutic, or ELBP. So the idea is that when a patient needs to take an antibiotic, we can transiently populate the gut with an ELBP that has the capacity of local sustained degradation of the antibiotic, so the microbiota is not exposed. And this protection can happen while the antibiotic circulates systemically in order to treat the infection that it was initially prescribed for. So that at the end, you can resolve the infection and end up with an intact gut microbiota. Now, there are multiple classes of antibiotics with different mechanism of actions. So we decided to start with one that are beta-lactam antibiotics, which are the most important class of clinically used antibiotics. In the US, they account for the 65% of the antibiotics, and they amount to 150 million prescriptions per year. So how do we engineer a safe beta-lactam degrading bacterium? We know that bacteria have evolved enzymes capable of breaking down beta-lactam rings called beta-lactamases. These enzymes are found mostly in gram-negative bacteria, and they locate to the periplasmic space where they can inactivate beta-lactams as they approach the cell wall before they reach their target. Under these conditions, beta-lactamases confer resistance to producer cells, and because of this increased selective advantage, beta-lactamase genes are readily spread via horizontal gene transfer. Of course, we don't want any of these features in our living biotherapeutic. We just want the capacity of inactivating the antibiotic. And this is why we took a synthetic biology approach to create a bacterial therapeutic that is able to secrete an enzyme to perform the extracellular degradation of the beta-lactam. So this activity would be beneficial for the entire microbial community in the surrounding environment and not just to the producer cell. In addition, the modifications that we incorporate have to maintain the susceptibility of to the antibiotic of the producer cell and avoid that this degradation trait can be passed to other bacteria through horizontal gene transfer. So how did we do this? We use Lactococcus lactis as a chassis for constructing our biotherapeutic. And lactis is a food-associated bacterium that humans have been safely consuming for millennia as part of fermented food products. Importantly, this bacterium does not colonize the human gut, but is metabolically active during its transit through the intestines, which makes it ideal for transient delivery of biological effectors. Also important is that there are genetic tools available so we can do engineering in this bacterium. So how can we make a safe beta-lactamase producing L-lactis? What we did was to refactor the biosynthesis strategy of the beta-lactamase to remove the features that confer producing bacteria a big selective advantage. We used a split gene strategy to eliminate the selective advantage conferred upon a single locus. For this, we divided the beta-lactamase into two fragments and encoded them in different genetic loci to reduce the chances of co-mobilization to a single potential horizontal gene transfer recipient. To reconstitute the enzyme and achieve extracellular beta-lactamase activity, we fused these fragments to cognate dimerization domains and tag them with extracellular secretion signals. In this way, once they are outside of the cell, they form a covalent bond that facilitates the restoration of the hydrolytic activity of the enzyme, and you regain the beta-lactamase activity. Now, what are the phenotypic consequences of this modification in the biosynthesis strategy of a beta-lactamase? Normally, beta-lactamase producing bacteria such as E. coli can survive the antibiotic action independently of their cell density. That means that a single cell can survive the antibiotic. In this case, we can evidence that in the formation of single colonies when grown in the presence of different concentrations of ampicillin. In the case of a lactis, expressing a split beta-lactamase system is very different. Since the beta-lactamase is not attached anywhere to the cell wall, it diffuses away, leaving the producer cell exposed to the antibiotic. To survive the action of the antibiotic, the population needs a threshold concentration where there are enough cells secreting beta-lactamase that can inactivate the antibiotic before the entire population collapses. Basically, the survival to the antibiotic becomes density dependent, and this is what we show here in this survival density threshold. Okay, so you might be wondering what would happen if for some reason, because there is a non-zero chance that both fragments would end up in a native host such as E. coli. To test that, we forcibly transplanted the SPTM1 system to E. coli and evaluated its survival phenotype. Just like with the lactis, E. coli expression of SPTM1 does not enable the growth from single cells and requires a high cell density to survive the ampicillin, likely because the pathway is inefficient in a different host. This demonstrates that the expression of the SPTM1 beta-lactamase system does not confer resistance to single cells, and therefore, at low cell densities, the producer cells do not gain any competitive advantage. To picture this better, we can put it in terms of the survival landscape to the antibiotic. In a sensitive bacterium, the split between the survival or death of the population is independent of the cell density and is dictated by the minimal inhibitory concentration. In a bacterium expressing a native beta-lactamase, this minimal inhibitory concentration shifts to the right, but the survival is still independent of the cell density. It doesn't matter if there's a million cells or just one cell, each one of those have the capacity of surviving to the antibiotic. In the case of our engineered live biotherapeutic, the survival is an emerging property of the population. At low cell densities, the cells behave as a sensitive bacterium, but after it crosses the critical threshold concentration, every cell in the surrounding environment will survive the antibiotic. Basically, below this survival density threshold, everyone dies, and above this line, everyone lives, which is very useful because you can control the behavior of the system based on the initial input of the dose. So to recapitulate this part, with these modifications, we are able to create a safe beta-lactam degrading bacterium by using a novel biocontainment mechanism that employs several evolutionary safeguards. It uses a food-associated bacterium that is gut transient. It provides extracellular beta-lactamase activity. Basically, it produces a public good for the benefit of the whole population, precluding any selective advantage in comparison to the other cells in the system. And it's encoded in a pathway that is proof to horizontal gene transfer. So you can think of this beta-lactamase as a shield that we don't want anyone else to use. When we split the shield into two pieces, it's not to make it harder to find, because eventually someone might be able to find two pieces. But what the splitting does is that even if someone finds the two pieces, they won't be able to tell it is a shield, and they won't know how to use it. So, well, this sounds good, but does it work in vivo? We used a mouse model to test the performance of the engineered-like biotherapeutic and the pharmacokinetics of the antibiotic. What we did was to populate the gut of the mouse with 2 doses of the engineered bacteria, one 2 hours before and the other at the same time with a single dose of intraperitoneal ampicillin. Then we collected blood, cecal, and fecal samples to compare this intervention with the untreated control. For these experiments, in mice treated with a lactis SPTM1, we detected an increased beta-lactamase activity in stool samples. Using mass spectrometry, we detected in situ degradation of ampicillin both in fecal and cecum samples. And despite this increased beta-lactamase activity in the gut, importantly, we did not find any significant differences in the concentration of ampicillin in the blood between the treatment and the control, which is fundamental to conserve the efficacy of the antibiotic treatment in infections outside the gut. This is gut, but is this activity enough to protect the composition of the gut microbiota? So to check this time in our model, we gave the mice a daily dose of ampicillin for 3 days. And for each one of those, we gave 2 doses of the engineered-like biotherapeutic as we did before. Then we collected samples throughout the experiment and analyzed them through a DNA sequencing to determine the composition of the microbiota. So basically what we're doing here is taking a picture before, during, and after the antibiotic treatment and see what happens with the gut microbiota. We first analyzed the bacterial diversity and observed that, as expected, the control mice that did not receive any protective intervention suffered a sharp drop in the bacterial diversity during the 3 days of the antibiotic treatment. And even after the antibiotic course was done, the diversity of the microbiota did not recover to the pretreatment levels, which speak to the long-lasting effects of the antibiotic that the antibiotics can have. On the other hand, the mice that received the L-lactis SPTM1 as the protective intervention suffered a small drop in the diversity that was reestablished to pretreatment levels even before the 3-day antibiotic course was done. We also compared the composition of the microbiota between different samples using principal coordinate analysis. You can see that before the antibiotic, all the samples clustered down here very tightly, and they are very similar to each other. After the antibiotic begins to have its effect, you can see that there is a divergence in the composition of the samples in both the control and the SPTM1 treated mice. However, for the mice that received the protective intervention, this digression in the composition is shorter than control mice, and they quickly recluster to the original composition of the pretreatment conditions, while the control mice never went back to the original composition. This result suggests that this protective intervention is effective in maintaining the microbial composition of the gut upon antibiotic treatment. But is this effect permanent? To test this, after 7 days from the last ELBP dose, we subject the mice in the SPTM1 treated and control groups to a second challenge with ampicillin for 3 days, but this time there was no dosing of any L-lactis or any protective intervention. We observed that the microbiota of mice in the SPTM1 treated group were susceptible again to the action of the antibiotic, as evidenced by a decrease in the Shannon diversity index and a disruption of the composition of the bacterial beta diversity in the principal component analysis. Then what is interesting is that mice that were in the control group, they did not suffer significant changes in their diversity after the second ampicillin exposure. It is possible that the lack of sensitivity to the antibiotic in this group is due to the presence of an extant population that is dominated by organisms that were selected for their capacity to stand the antibiotic during the first ampicillin course. These results underscore the type of perturbations that antibiotic can cause in terms of the composition and the susceptibility to the gut microbes, and shows that our treatment can maintain the diversity and composition of the microbiota overall at the DNA level. Now, in antibiotic-induced dysbiosis, the concern is not only about the microbial groups that are lost, but also the ones that are able to remain and increase in their abundance. Antibiotic treatment does not have a sterilizing effect in the gut. Instead, it selects for population that encodes some sort of resistant mechanism. And when these populations expand due to their increased fitness, compared to the other bacteria that die with the antibiotic, the gut becomes enriched in antimicrobial resistant genes. Since the microbiome is truly a network of genes, this enrichment increases the chances of transferring these resistance mechanisms to potential pathogenic bacteria. And now it is becoming apparent that this type of within the gut horizontal gene transfer events are contributing to the emergence of new antimicrobial resistant pathogens. So we evaluated how our intervention might affect or might help, with this issue by detecting and quantifying antimicrobial resistant genes in our mouse model. We observed that before the antibiotic treatment, there was only a few genes of the different resistant classes, but upon antibiotic treatment, as expected, the mice in the control group became enriched in genes related not only to beta-lactam resistance, but also to other unrelated classes of antibiotics like glycopeptides, non-ribosomal peptides, and as well as efflux pumps. In contrast, despite the ampicillin treatment, the presence of the L-lactase that encodes the SPTM1 beta-lactamase significantly reduced the expansion of the antimicrobial resistance genes from the endogenous pool of the gut microbiota, which means that the intervention can effectively reduce the selective pressure of antimicrobial resistance genes. Now, using the same approach, we can evaluate the selective pressure on engineered and endogenous beta-lactamase during the intervention. Notably, despite having fed the mice more than 10 to the 10 CFU lactase SPTM1 cells during the 3 consecutive days, the SPTM1 beta-lactamase fragments were present only in trace amounts by 3 days after the last ELVP dose and undetectable by 5 days. This effective depletion of the large pool of the engineered beta-lactamase fragments concerns with the strong enrichment and selection of endogenous beta-lactamase genes that emerge from a small pool in the control group during the first ampicillin course. What is more important is that upon ampicillin rechallenge of the ESPTM1 treated group, there was a significant enrichment of endogenous beta-lactamase genes. The microbiota is susceptible again to this, but no SPTM1 beta-lactamase gene fragments were detected, suggesting that any lingering engineered fragments from our beta-lactamase were not under the selection of the antibiotic despite its pressure. These results reinforce our claim that the expression of the SPTM1 system does not confer the added selective advantage to the producer cells in vivo, so that our design in which we are having a horizontal gene transfer proof pathway is supported by these data. Now, up to this point, we have observed the protection of the gut microbiota at the DNA level, but does this translate to a clinically relevant outcome? We wanted to provide a functional demonstration of the protection, and we use colonization resistance against C. difficile as a readout. A fully assembled diverse microbiota can keep C. diff away by means of metabolic competition. Exposure to the gut microbiota to antibiotics eliminate the commensal bacteria from their intestinal niches and allow C. difficile to colonize and proliferate. So in our protective intervention, if our protective intervention is effective at preventing the collapse of the gut microbiota, we should be able to see a maintenance of the colonization resistance even after the treatment with ampicillin. So we used our mouse model. We gave the mice the 3 doses of ampicillin and their corresponding L-lactis doses. And the day after the 3rd dose, when the microbiota has been exposed to the antibiotic for the longest, we challenged the mice orally with C. difficile spores. Then we collected fecal samples at 24 and 48 hours post-infection to detect possible colonization. Remarkably, all the mice that received the beta-lactamase expressing L-lactis did not develop a C. difficile infection by 48 hours post-infection, while all the animals in the control group that received the L-lactis carrying an empty vector developed a full-fledged infection, indicating that the mere presence of L-lactis is not enough to provide colonization resistance. These results demonstrate that the L-lactis SPTM1 protects the mouse gut microbiota from ampicillin to an extent enough to preserve colonization resistance against C. difficile. Of course, in this experiment, we also determined that there was an increased beta-lactamase activity in the gut, and despite the 3-day treatment with the genetically engineered bacteria, the levels of ampicillin in serum were not modified compared to the control, which is still important to, again, maintain the efficacy of the original antibiotic. So this work provides proof of concept on how an engineered live biotherapeutic can be used to prevent antibiotic-induced dysbiosis. In the experiments I showed here, using a mouse model of beta-lactam therapy, support the notion that this ELBP is safe and biocontained. It can preserve the composition of the gut microbiota. It prevents C. difficile infection and reduces the enrichment of antimicrobial resistance genes in the gut. We think this is a promising technology, and we're beginning to explore how we can use it to demonstrate a clinical benefit in preventing antibiotic-induced dysbiosis. So what is the landscape? Basically, every patient in need of a beta-lactam antibiotic is at risk of dysbiosis. Now, there are a subgroup of patients that, depending on some risk factors, are more susceptible to developing certain types of diseases. We know that pediatric patients that are constantly exposed to antibiotics are at higher risk of developing obesity or asthma. Patients prescribed with antibiotics that have comorbidities such as obesity are at higher risk of developing IBD or diabetes. These are risks that are well documented, but the problem is that these diseases manifest in the long term, and having a clinical valid readout to demonstrate efficacy is in a reasonable time frame is difficult. So it is more reasonable to focus on diseases related to antibiotic-induced dysbiosis that manifest in a shorter time frame. Of course, opportunistic infections by C. difficile, primary C. difficile, or vancomycin-resistant enterococcus are good leads for this. Also, we are exploring how we can use this technology to increase the efficacy of certain cancer treatments, such as stem cell transplant and immune checkpoint inhibitors, for which we know that the preservation of the gut microbiota is critical for a positive response to the treatment. Also, in general, This technology can be adapted to be used as an additional antibiotic stewardship effort in order to reduce the emergence of new antibiotic-resistant variants stemming from the gut microbiota. If you have a way of demonstrating the clinical efficacy of this approach like this, we are open to collaborations, and we are eager to show how we can benefit patients for this. So before I finish, the message that I want to leave here in the context of this Microbiome First Summit is that we need to protect the gut microbiota like we protect other major organs. Whenever we're going to be exposed to UV radiation from the sun, we apply sunscreen to our skin to prevent DNA damage and possibly cancer. When a patient needs an X-ray, a leaded apron is used to protect the rest of the body from the harmful action of the ionizing radiation. Now that we are realizing about the importance of the gut microbiota, we need to start protecting it from the damaging effects of antibiotics. And this is an important unmet need that is fundamental if we really want to have sustainable healthcare. And with that, before I finish, I want to thank the people involved in this work, especially Professor Jim Collins. This technology has been developed in his lab under his guidance and support. I also want to thank the rest of the members of the Collins lab at the VIS and at MIT, especially Miguel and Nina have been instrumental for the development and validation of this technology. And to our founders, DTRA, the Allen Foundation, the Center for Microbiome Therapeutics at MIT, and especially to the VIS Institute that are supporting the translational efforts of this work through a VIS validation project. Thank you for your attention.